outbreeding devices. So, what are outbreeding devices? Well, plants have many mechanisms and devices that are employed to promote cross-pollination, and these are called as outbreeding devices. These devices discourage self-pollination and encourage cross-pollination. But why? What is the reason behind it? Well, this is because continued self-pollination results in inbreeding depression. That is, the decrease or loss of fitness and strength, which is mainly caused due to inbreeding. Some outbreeding devices are: number 1, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of the pollen. Number 2, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. Number 3, the third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self of pollen from the same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Number 4, the fourth device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and monoecious, it prevents autogamy, but not geitonogamy. While in other species, such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, dioic. This condition prevents both autogamy and geitonogamy. Can you imagine how dull our world would be had there been no variations? Our future flies on the wings of pollinators. So, this calls for some serious attention towards our next topic, which is pollen-pistil interaction. As the name suggests, pollen-pistil interaction tells us about the pistil accepting the right type of pollen. Interesting, right? Pollination does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Often, pollen of the wrong type, either from other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, compatible, or of the wrong type, incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that lead to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. So how does this happen? The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of the continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. So, what happens next? Well, after compatible pollination, the pollen grain germinates on the stigma of the pistil and produces a pollen tube through one of its germ pores. The pollen tube grows through the tissues of the stigma and style and reaches the ovary. Now, through this pollen tube, the contents of the pollen grain are transferred to the ovary. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of these synergids through the filiform apparatus. The students, all these events from pollen deposition on the stigma until the pollen tube enters the ovule are together referred to as pollen pistil interaction. So, what happens after the pollen tube enters the synergids? Well, now the process of fertilization takes place. But how? Well, after entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, PEN. As this involves the fusion of three haploid nuclei, it is termed triple fusion, since two types of fusions, syngamy and triple fusion, take place in an embryo sac. The phenomenon is termed double fertilization, an exclusive feature of the flowering plants. You might have heard of the artificial hybridization, right? So, what is artificial hybridization? Well, artificial hybridization is the process in which only desired pollen grains are used for pollination and fertilization. Yes, a plant breeder can manipulate the pollen pistil interaction or even incompatible pollinations to get desired hybrids. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination, and the stigma is protected from contamination from unwanted pollen. But how? Any guesses? Well, this is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehisces using a pair of forceps is necessary, and this step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. And this process is called bagging. This ensures that the flower is pollinated by pollen grains obtained from desirable varieties only. When the stigma of bagged flower attains receptivity, mature pollen grains collected from anthers of the male parent are dusted on the stigma, and the flowers are rebagged, and the fruits are allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen, and the flower rebagged. Now let us sum it up then. In this module, we learned about pollination, kinds of pollination, agents of pollination, outbreeding devices, pollen crystal interaction, and double fertilization. By now, we all know that pollinators are often keystone species, meaning that they are critical to an ecosystem. The work of pollinators ensures full harvest of crops and contributes to healthy plants everywhere. For just this to happen, a marvelous bond exists among different natural elements. Humans here as well manage to use their intelligence and create something new and hybrid. Despite the fact that they make possible the life for us, their lives are in peril. Do you know that it is estimated that nearly 40% of them may face extinction in the coming decades. So, it becomes very essential to protect these pollinators in the world of pollution and climate change. By doing this, we will not just save their lives, but our lives as well. It is impossible to imagine our world without pollinators. The students thank them and meet me in our next module to understand how amazing work they do. Yes, we will learn about the post-fertilization events in the flower. Hey, why don't you find some pollinators in your garden? Make sure the next time you see them, bees, birds, butterflies, please let them in and watch the magic unfold.